Hi, Daphne. This is Steve Hsu. We're going to talk about some things related to genetics of cognitive ability and the impact on education and education policy. Maybe you can just introduce yourself.
Yes. Well, thank you for speaking with me. I'm Daphne Martschenko. I'm a PhD candidate at the University of Cambridge in the Department of Education, and my research looks at the implications of behavioral genetics research on cognitive ability, educational attainment, academic achievement, on the United States education system in particular. And so I had some questions kind of about the science behind the academic journals that I've been reading, and the book G is for Genes, published by Catherine Asbury and Robert Plomin, which I really center a lot on within my work. So one of my first questions was kind of understanding the difference between a polygenic score and a genome-wide polygenic score. There was a recent study published out of the TEDS group in the UK, CELSM, and they use genome-wide polygenic scoring and mention it as a kind of new analytic tool. So I'm kind of looking to see what the difference is between what was used before.
Okay. So I might not know exactly the difference between these two things. I can tell you what I think of when people say polygenic score, and I think I can say a little bit about what the current state of the art is for this. So in these genome-wide association studies, you have a bunch of data where you have the individual's, say, cognitive score, and then you have their genotype. Mm-hmm. And what you're looking for is statistical associations between a particular gene variant and their score. And if you find that people with variant A at a particular locus in the genome, so a particular SNP, if people have one variant of it, they tend to, on average, just be slightly higher on the score, and people who have the other variant tend to be slightly lower on the score. If that is statistically significant, if the effect is strong enough, the analysis will say that particular SNP is implicated as being associated with cognitive ability. Okay? And furthermore, the method will estimate an effect size. So on average, how much, quote, smarter or whatever, how much will the increase in score be for having version A versus version B? So that's the effect size. And some SNPs will have larger effect sizes and some will have smaller effect sizes. Okay? And I believe the current state of the art is they've found maybe a few hundred SNPs now, which pass the most stringent tests of statistical significance. So it would be fair to call those SNPs or genetic variants that are associated to cognitive ability. And if I wanted to compute an individual's cognitive score, what I would do is I would just simply look at that particular individual, look at the first of the 300 variants and say, well, do they have the plus version or the minus version? If they have the plus version, I increment their predicted cognitive ability by whatever the effect size is. I then go to the next one. If they have the minus version, I decrement their predicted score by a little bit. So it's basically just adding up all the effect sizes that have been deduced from the previous analysis, but specifically for an individual. And so you get a different polygenic score for different individuals. And some people might have an unusual number of the plus versions. Some people might have an unusual number of the minus versions. In one case, that person would be predicted to be well above average. And in the second case, that person would be predicted to be well below average. Now, there are all kinds of mathematical subtleties for what is the best predictor, how to construct it. Should I just use the effect sizes that I just talked about, or is there a more sophisticated way to do it? If you have some gene variants that fall a little bit below the statistical threshold in order to be included in that few hundred, should I also include them in the predictor? Because very likely they are actually associated with cognitive ability. Maybe 1 out of 10 of the ones that are on the edge maybe are false positives, but I could gain some predictive power by including those guys. So those are all sort of kind of mathematical details for how to actually build the best or optimal predictor. And so one could talk about different kinds of polygenic scores. Poly just means many, and so it's a score that depends on the input, which is many, the state of many different genes or different SNPs in a person's DNA. Does that help?
Yes, it does. Thank you very much. And another methodology that I've seen is the candidate gene design, which I've read is often fails to replicate in a larger sample. Could you explain perhaps why candidate gene design might still be used over the polygenic scoring?
So if you go back like 10 years when all these genetic association studies were quite new, and at that point, it was much more expensive to get the data. So it was much more expensive just to get a sample size of like 1,000 people. Whereas now we routinely have sample sizes like 100,000 people. In the beginning days, what would often happen is some biologists, and this extends not just to cognitive ability, but it could be health or disease risk or whatever. Some biologist or doctor would say something like, well, I know this gene codes for this protein, and I know this protein is really important in the pathway that, I don't know, builds your eye or something, okay? And so that person, based on their medical or biological intuition, would say, I think there's a very good chance that this specific gene, which codes for this specific protein, or this, gene could be contain many individual base pairs. So it could be, well, this specific mutation on this gene or this other specific mutation on this gene. I have some reason to suspect that that will be associated with how well your eye functions, like how susceptible you are to some eye disease, okay? So that in the Bayesian probability language, that's called a prior. I have some prior belief, it's informed by my medical expertise or biological knowledge. And because of my prior, I'm pretty confident that the genetic variants that are on this specific gene are going to turn out to be somehow associated with your risk for this eye disease. And people proceeded that way 10 years ago, and they would do studies which were very underpowered. And they were allowed to publish results in which, okay, maybe they got a slight elevation in the risk. So people were just slightly more likely to have the disease condition if they had that suspected mutation, some version of it, okay? But the statistical certainty was allowed to be quite weak, so p less than 0.05, okay? And so what people were mainly doing is they were trusting their biological or medical intuition, and then they were just allowing fairly weak statistical evidence to then convince them or convince the journal editor or the referee that indeed we've discovered an association. In the modern era, we don't do that. We actually don't allow you to pre-select like, oh, these are the genes that I'm almost certain are going to be the ones that affect the disease. We actually look at the whole genome, and we require a much, much stronger statistical threshold, p less than 5 × 10⁻⁸. So it's 1,000,000 times stronger, this statistical requirement, and of course requires much larger samples. So now, it could have been the case that people's intuitions 10 years ago were really spot on. And in fact, when they trusted their intuition, they ran a weak statistical test, and the candidate gene passed that weak statistical test, that then further research would show, oh, that's correct. The result replicates. When we get more power, we run it again, and we also still find an association between the mutation and the disease risk. But it turned out the other way around. It turned out that as we got bigger and bigger data sets, almost none. In fact, a famous guy at Stanford called Ioannidis, who studies the scientific method, he claims to have done some sort of backward-looking analyses of candidate gene studies, and he says like less than a few % replicate. So literally what that means is if you look at journal articles published in the most prestigious scientific journals between like 2005 and 2010, I'm just making those dates up, but that was kind of the rough time. And you looked at all the candidate gene results that, oh, my lab at UCLA got this, and my lab at Michigan got this, and my lab at, only a few % of those turn out to be true. So it's that was a very bad technique. And the lesson I think people should learn, which effectively they've learned because the way we do things now is with this 5 × 10⁻⁸, much stronger statistical threshold before you can publish the result. But the real thing you need to learn is that the priors that people had, that they were very confident in, were not very good. So people thought they really, I have a kind of handle on how this disease works, and so I just need a small amount of additional statistical evidence, and then I'll believe that this mutation is really playing an important role. But it turns out not to be true. People don't really understand. Biology is just too complicated, and it's better to just do brute force things with huge samples and just trust this statistical analysis rather than trusting human intuition. I think that's a lesson from the candidate gene era. Now, I should add that in the modern era, once you find a hit, an association which passes the p less than 5 times 10 to the minus 8 threshold, it routinely replicates. So some other group, which might be working with, Asians or people in Africa or whatever, if they then check that statistical association, they will find it as well. And so the new standard seems to be a good standard, and it seems to actually produce results that routinely replicate.
Twin study.
Oh, and by the way, when I said, for the polygenic score, they have like some number, a few hundred hits now or something like this, or 100 or plus hits, those are according to the new stricter standards. So we expect that they're real.
Okay. Also, twin study is used a lot for these genome-wide association studies.
Yep.
And I've read that twin studies indicate the maximum possibility of heritability. And, coming out of twin studies, there are fairly reasonable heritability estimates for cognitive ability using twin study.
Yep.
And so I'm wondering whether the missing heritability problem or the issue of not necessarily finding markers that add up to the heritability estimates from twin studies could be a reflection of the fact that twin study design is actually estimating an amount that's higher than reality. So
yeah, it's entirely possible because, so in a twin design, you have, say, 2 identical twins, and maybe they're raised in different families, and then you look at how similar they turn out to be, say, for cognitive ability or IQ score. And from that kind of analysis, some people might claim heritability is as high as like 0.8 or something like this. And but there's still many confounds. It could be that although we raise these twins in different environments, so we think we've randomized over the environments that the 2 individuals have experienced, it could be that because they look alike, they're treated alike. Even though this family doesn't know this family, you've got 2 individuals who look very similar to each other, and maybe there's some similarity in what they experienced, even though they never met each other or anything. So it could be that 0.8 is an overestimate. I think very few people would think it's a super overestimate, like maybe instead of 0.8, it's 0.7 or it's 0.6. But very few people would assert that it's like 2 times too large or something like that. Mm-hmm. Now, the current situation with the genomic predictors and polygenic scores and such is that they can account for some chunk of variance, but that chunk is still small relative to 0.8 or relative to 0.7 or relative to 0.6. And so there's a gap. And so people often refer to that gap as missing heritability. Some of the gap closing may be due to the original number being overestimated. Like maybe you don't need to really get to 0.8. Maybe 0.8 was inflated and you really just need to get to 0.6. I think most of the gap, though, between our current ability to do genomic prediction and where the ultimate limit will lie is mainly just due to statistical power. And my prediction is that once we get data sets where we have like 1 million people in the data set, not 100,000, but 1 million. So right now we're kind of at like the 100,000 level. But when we get to like 1 million, my prediction is we'll capture most of the implied predictive power that's in a heritability of 0.7 or something like that.
My next set of questions kind of deal with gene-environment interactions. I have noticed that there have been a number of studies that kind of look at the relationship between cognitive ability and socioeconomic status. And
yep.
There seems to be a consensus that heritability estimates for cognitive ability are higher for individuals who are brought up in higher socioeconomic environments.
Yep.
But when I was reading G is for Genes, they also talk about how socioeconomic status is partly influenced by genes and also correlated with IQ. So I guess my main question is, how do these studies kind of distinguish between socioeconomic status as nurture and socioeconomic status as nature, or whether they even do that? So I guess how can it be a gene-environment interaction if the part that they're looking at to be environment is itself partly influenced by genes?
Yeah, so you're hitting on really one of the most difficult aspects to this research. And it'll take, I think it'll take a long time before all this is settled. If you're on the genetic side and your main interest is just finding the genetic architecture for these traits or whatever, your kind of gold standard would be like, oh, this million sample size that I was talking about. It would be great if all of these million people came from sort of good SES background. So all of them had access to good schools and trips to the library and all that stuff. So you could try to minimize as much as possible the effect of the environment. There have been studies that show that if people are deprived, like they literally, they go to crappy schools, their parents never take them to the library, they don't have any books in the home, that they won't achieve their genetic potential even if they have high genetic potential. And so that you have this big effect maybe from the environment that screws up your ability to do the genetic study. So it's going to take a while to disentangle these effects. I mean, obviously the ideal thing is to only look at people who come from really favorable environments, and then the heritability is high because they all have good environments, and the main reason that person A is different from person B is really due to their genes. That's the ideal situation. Now, one of the things you mentioned just now is that when you have somebody who's born in a low SES family or a high SES family, it's true that they then have a different experience in their education, right? More books, more tutors, fewer tutors. But it's also true that systematically it could be that the reason their parents are in the high SES group or the low SES group is that their parents themselves could be above average in their genetic potential. And that's an additional confound. So it could be like, well, the high SES kid is doing well. Part of the effect is the parent could invest more in education. Part of the effect is that on average, the people who are successful in society already had good genes. And so you have to disentangle those two effects. It's quite hard. There's a study which I can send you a link to recently, which used the polygenic scores. And it was a group in New Zealand, and there's a famous cohort study called the Dunedin, I guess that's a city in New Zealand, Dunedin study. And they actually looked at the polygenic scores of different groups of people, and they found actually that the higher SES people tended to, on average, be shifted slightly to the right on the polygenic score, and the lower SES people were shifted slightly to the left. So there's some evidence that part of what helps you succeed in society is your gene, and so you have this your genes, and so you have this additional confound that the high SES kids are doing better, partially because they got better genes from their parents, but partially because they got better environments from their parents. So it'll be very tricky to disentangle all these effects in the future.
And I guess I personally saw, because there seems to be a little bit of a conflict between the social and the hard sciences, particularly in this area. So I at least saw the gene environment studies as a kind of attempt to bridge the argument that social scientists make, which is that environment matters. And I'm wondering whether other gene environment studies have been done in relation to cognitive ability outside of socioeconomic status. For example, I know that there's some debate over whether self-identified race should be used in genomics research. Some saying that it's valuable, some saying that it's not a legitimate biological construct and should not be used. But I'm wondering whether self-identified race at all or any other environmental aspect has been examined in relation to cognitive ability.
Well, going back to your earlier remark that there's a little bit of conflict between the, quote, hard sciences and social sciences, I don't think it's true that the people trying to do the quote, hard science, the genetic aspect of it, want to disregard environment. In other words, almost everybody would talk about it kind of the way I talked about it just a few minutes ago, which is saying that, you know, the environmental variations are a confound, and they're one that we're trying to avoid because we want to get accurate, correct results. And so the people on the hard side, you know, like I said, oh, what would your ideal sample be? My ideal sample would be 1 million people, but all of whom came from, you know, very good backgrounds, and and nobody had a bad environment. Everybody had a good environment. In fact, ideally, they'd all have very similar environments. So we're obviously cognizant of the environmental factor. We're trying to minimize it so we can get at the part that we're researching specifically. As far as race, now, what's the right way to say this? So the way we talk about race, it's a very complex subject. So I understand why people say it's a social construct. So if you say, like in the US, we have this one drop rule, right? So if you had a little bit of African-American ancestry, people would say, oh, you're black, which is obviously a very imprecise way to talk about stuff, right? However, if you do look at people who trace most of their ancestry to very different places, like some guy traces most of his ancestry to Japan, some other guy traces most of his ancestry to England, there will be systematic genetic differences. So if you look at a particular SNP or particular variant, you could have a situation where 80% of British people have variant A, but only 10% of Japanese people have variant A. And if that variant turns out to be one of the ones that's actually affecting cognitive ability, then you could have an effect which builds up between the two groups and is important. So I think people mainly don't want to include race as a variable in all this because it's very politically charged. And, you know, we were talking earlier about environment being a confound. If you have a society where people are treated differently based on their ethnic background, that's a huge confound, right? So when I said, oh, my ideal sample would be 1 million people, all from good socioeconomic backgrounds, I would also like them all to be from the same race, because if, say, half of them are Chinese and half of them are white, or half of them are African-American and half of them are white, they may have been treated very differently growing up. And so that's a yet additional confound that I just would rather not deal with. So my ideal samples would always be take a bunch of people, all who come from the same place in terms of ancestry and have similar SESs, and then we'll look at the effective genes within that group. What makes one British guy smarter than another British guy, or what makes one Japanese kid smarter than another Japanese kid. That's much easier to get at than cross-racial or cross-ethnic things because of these additional systematic uncertainties.
I also get the sense that perhaps some people are not as open to talking about race in the manner that you've just done it, by that is to say, by engaging in the conversation at all. And there has been, in the social sciences, some discussion of perhaps the danger of not even talking about race in relation to this work, simply because of the history that underpins the field. So I'm wondering whether, given the history that underpins genetics research as a whole, and then more specifically intelligence or IQ, whether it is valuable or meaningful to discuss race, even if only to say that this work can't say anything with regard to race.
I think for me, the best statement to make right now is we should be extra careful about any conclusions that we jump to regarding race. Because I said the ideal study would be you have a sample and they're all from the same ethnic group, and then you can make some conclusion, and the conclusion would be something like, oh, I can construct a polygenic score. This score was specifically constructed using Northwest Europeans, and I have some ability to predict from the genome of an individual Northwest European what his or her cognitive ability will be. It would be a big leap to take that predictor, the specific set of gene variants and the specific set of effect sizes or whatever, and try to apply it to a bunch of Chinese people. It would be extremely risky because there are a bunch of factors that could go wrong. Mm-hmm. And so that should be a step that's taken much later, after we have, we've done individual studies of Chinese all by themselves, and we have an understanding of whether the individual SNPs are tagging the same variants in the actual genome across the both ethnic groups. There are a bunch of issues that would just need to be resolved. So I would say it's very early days, and the best thing we can try to do is study one particular group of people in a very clean way. And then there are results that you can, I think, trust. Like, you could eventually get to a polygenic score that does have some predictive power for a particular ethnicity or ancestry group. But I would be very, very careful before trying to generalize that polygenic predictor to other groups.
And correct me if I'm wrong, but I believe most genome-wide association studies done on cognitive ability deal predominantly with European ancestry population samples.
Yes,
So,
that's correct.
So my question was just, are you then saying that findings of those studies cannot necessarily be applied to other ancestral groups?
Yeah, I think you have to be very careful with that because, well, there's a very technical reason, which I can just quickly summarize, but it's kind of for experts. When you measure the genome of an individual, you measure the value of a particular SNP, okay, whether it's like GCTA at this particular location. And what that letter of the DNA alphabet is tagging could be some really complicated genetic level difference. Like if they have a G here, it's associated with them having some big blob of mutation in their DNA. And if they have the other letter, they have something else. But that association could be specific to a particular ethnic group. It could be that the difference between letter G and letter A for another ethnic group means something different when you actually look at the underlying DNA. So remember that a SNP genotype, which is very inexpensive to obtain, you can genotype somebody for like $50 or less. That's just sampling from about 1 million places on the genome, and it's not the same as a whole genome sequencing where you get every location. And so the meaning of a particular SNP variation could be different for different ethnic groups. And so even that by itself is enough of a uncertainty that you would have to think really hard about that before you try to apply your polygenic predictor constructed on one group to another group.
Thank you. My next questions kind of center predominantly around IQ tests. I know that IQ tests are often used as a measure for cognitive ability. And again, they're on predominantly the social sciences side, there is some critique of the value of IQ tests. For example, I've read that there's been a history of manipulation of past scores on IQ for certain groups. And also there's the constant renorming of IQ scores, which means that there's always going to be a group at the top and another one at the bottom, but that won't necessarily tell us the value of their abilities. So my first question is, how reliable are IQ tests as measurements for cognitive ability?
So I think the best large data set to think about in this context is things like SAT and ACT scores. And so there are many results, like if you take a kid and you give him the SAT and then you even let that kid prep, like you let them go to Kaplan's and do a bunch of stuff, and then you test them again, and you could test them again quite a long time later, like 6 months later or a year later. And you can ask, well, how much does the score fluctuate? Like, how much do they really gain from all this test prep, or if they study really hard for it? It's generally a relatively small difference, like 50 points or 100 points. So if you have two individuals, I'm from the old era where the ceiling was 1600 because we didn't have the writing component, so 1600. But you could have a kid who scored 1500, which is quite high, and you could have another kid who scored 1200. Okay? It's very unlikely that if you retest the 1200 kid, he or she will ever get close to 1500. And even after they've taken Kaplan's or whatever, despite anecdotal studies which claim that there can be big score changes, the test prep companies like you to believe that. But if you look at systematic academic studies, which have been done with thousands and tens of thousands of people, the stability of the score is quite high. So in other words, you test the kid a year later, and maybe they even did test prep during that year, their score might change by 100 points, but a change greater than that would be quite rare. Okay, so the value of the score is pretty stable. Okay, 1200 kid is actually different from a 1500 kid, usually. Now, what can you predict? Like, say you have a bunch of 1500 SAT kids and a bunch of 1200 SAT kids. What can you predict about, say, how they'll perform in college? Like what their GPA will be, how many of them could get through a really difficult major like electrical engineering or something like that? Well, the test does have pretty good predictive power. And there are really large data sets available for this. So there are universities that have basically made available for study not just the grade point averages, but the grades in every single class for a huge cohort, tens of thousands of students, and their SAT scores and their high school GPAs. So we actually know a lot. Like if you're a data wonk, you could make very precise statements like, oh, SAT score correlates 0.5 with upper division GPA. You could make very precise statements about how much predictive power you get from the tests. And I would say they're very valuable tools. I think schools that decide not to use the tests and admissions are making a huge mistake. They're not going to be able to have as much predictive power in terms of who will do well at the university and who won't if they don't have test results like this.
And what would you say to the comment that IQ tests have certain cultural biases? Do you think that is?
They certainly could. So in the early days, like you would have very specific vocabulary scores, for example, that, one group of people might know, would be much more likely to know that vocabulary than other groups. But now the set of IQ tests is quite broad. And so there are some that are very, can you hear me? Hello? 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 Hello?
Okay.
Sorry about that. Even in the 21st century, we can't have a stable. I'm sorry, what was I saying? You were asking about cultural biases. So, but some of the IQ tests are very, I think, culture neutral. Like you can have, there's a type of test called Raven's progressive matrices in which just show you some patterns and you try to figure out like how the pattern is evolving in time and guess what the next snapshot will look like. And it's hard for me to believe that that's very culturally biased. It could be there's some subculture of people who play with those puzzles all day long, so they're really good at them. But by and large, I don't think there's a huge cultural bias. Now, the SAT, which I used as an example earlier, obviously like having a good high school education would make you better at the SAT, right? So if you just had a crappy education from day one and you didn't read very well and you didn't learn basic math, you're not going to do well in the SAT. So there's some basic requirement that before you could say this is an unbiased measurement of somebody's cognitive ability.
I'm going to skip over the next section and go to the one on genetics and education, if possible,
Okay.
Since we're a little pressed for time. But again, since I am coming from the education department at the University of Cambridge, this part is really very interesting to me. And there are, I've increasingly seen people discussing the potential benefits that genetics research might have for education policy. So I just wanted to briefly get your point of view on how important genetics research on cognitive ability is for education policy.
So, if the idea is to like figure out which kid has high cognitive ability and which kid maybe has below average cognitive ability, in a way, it's like, well, why do you need to measure their genes? You can just measure them, right? You can just give them the test, make it as culture neutral as possible, and just see which kids are good and bad on the test. In the same way, like if you wanted to know genetically, like which kid is going to be a good runner, why don't you just have the kids run a race and see who actually runs faster, right? Do you really need to like measure their genomes? So I think that's the hurdle that the use of genetics in this context has to get over. Like if I have direct access to the person and I can measure their algebra ability, why do I need to like look at their DNA, right? Mm-hmm. Now, I think Plomin and Asbury they've proposed some scenarios, right, where I think these things could be valuable. There are scenarios like imagine you have a kid and you could have a kid who the polygenic score is extremely high, but the kid is not doing well in school and not doing well in the test. So you might have a case, like a kind of exceptional case where you have reason to suspect this kid has a lot of potential, but there's some reason why they're not doing well in school. They don't like school or they have a behavior problem or something else. So you could imagine situations where the genetic test would suggest to you this kid has some potential and we should focus on this kid, even though the actual real world performance isn't good, and vice versa. So you could have a case where the kid maybe has a very low polygenic score, but they're doing well. And you might say, wow, this kid is really outperforming. Maybe we should study what makes this kid so good, even though, on the genetic side, they don't seem that promising. So I wouldn't say there are no uses, but I think it's going to be a long time before we get to the Gattaca world where they just read out your DNA and they say, okay, you go to Harvard and you go to Northeast State University. I think it's going to be a long time before we can do stuff like that.
Mm-hmm. And there also there are just differences between genome-wide association studies on IQ and then academic achievement and educational attainment. And I've seen all three.
Yep.
And I'm wondering whether one of these areas might be more beneficial for K through 12 education than another.
Yeah, I think
how three might differ, I guess.
So a lot of times what is actually used as the phenotype variable in the study is by convenience. It's what they can get their hands on. There are differences between those phenotypes that you just mentioned. So if you think about it, like we all know people who are really smart, but they're super lazy. And we know people maybe who aren't that bright, but they're very organized and they do well in school. So you can see that the grade point average or how far you get in school, it depends on multiple factors, not just your raw intelligence. And so probably what these school achievement things where you actually measure like how far did you get in school or what was your grade point average, generally you're measuring some combination of things, your raw cognitive ability, but also like your conscientiousness or your organizational skills. And so, but it may turn out that for predicting success in life, all of those factors are important too. So it just depends on what you're trying to do with the polygenic score at the end.
So NGs for Genes, Asbury and Floman clearly are advocating for using genetics in education, curriculum, and policy. And they acknowledge that, the technology and the research is not yet at a stage where we can do that, but that we should start having conversations now about potential benefits or challenges for what they are proposing. And Ben Domingo out of Stanford also is advocating, he's associated with the SSGAC, but he's also advocating for the potential benefits of genetics research in education. And he says, and this is a quote, that we soon might be able to ask questions about whether certain students are more or less likely to respond to an intervention given their genetic profile. And also that it could be in the near future, we see parents, for example, demanding interventions for children based on their genetic profile. And you've talked a little bit kind of already about the feasibility of this, but do you think that there will be a stage when, in fact, that is possible? That what I mean by that is to have interventions that are known to be most effective for children with certain genetic profiles.
I think in the long run, sure. All these things are possible. Okay. I mean, at the moment, it might be a little bit like science fiction, but eventually you might find you might have a very kind of nuanced cognitive profile for somebody based on the genes. And you might say, well, this kid really could do with more interaction in their learning process. Or no, this is a kid who just needs to be left alone in the library to read. People have different learning styles. And it's possible in the long run that you could, quote, figure that out from the DNA. I think it's difficult. Like right now, I would just say the challenge is, can you build like a decent genetic predictor for very crude variables like total IQ or abilities to succeed in school? And I would be, I would be very pleased if in the next 5 years we were able to construct those predictors. But in the longer term, we might be able to do better than that.
And as Asbury and Floman mentioned, it's important to have these conversations now to kind of tackle some of the ethical concerns that might arise out of the future possibilities of this research.
I totally agree.
I just wanted to get your comment on that.
Well, one scenario which I think is worth mentioning that I didn't mention yet is, there's a big push to want to compensate teachers based on the actual rate at which their students are learning. So you give them a test at the beginning of the year, you give them a test at the end of the year, and you see how much the students improve. And maybe this teachers whose students do really well, they get a bonus. Okay. But if you want to do that really fairly, you'd want to know what the genetic potential of those kids is, because you might be rewarding the teacher for just having very talented students. And maybe the teacher's not actually a better teacher. So, even to be fair about that, like incentive structures for teachers, you might want to have some idea of the quality level of the students, and maybe the genetics could inform that. So you can imagine applications like that. I totally agree with them that this should all be discussed, because if you think back, not even that far back, but just, even now, I think there are a lot of people who are opposed to the ideas that genes play a role in all of this. They just don't like the idea and they don't want to acknowledge it, but the science is moving ahead very quickly.
Those were my list of questions. I know we're almost out of time, so I wanted to thank you and ask if you had anything that you wanted to add or questions for me.
I just want to encourage you to keep working in this direction. I think this is really important stuff, and it really could use somebody who can bridge the gap between the people that are doing the frontier research and people who are educators and trying to make use of the results.
Thank you so much for taking time out of your busy schedule to speak with me. I really appreciate it.
Yep, my pleasure.
Thank you.
Okay.
Yeah, that's it.
Okay.
Have a wonderful day.
Okay.
Bye.
Bye-bye.